Welcome back to the physiology of the gastrointestinal tract. My name is Ayman Saik and I teach at Tuskegee's Vet School. Last time I talked about the importance of understanding gastrointestinal physiology. Uh, then I talked about the definition of the gastrointestinal tract. We said it's a tube-like structure that extends from the mouth to the anus and provides the body, including itself, with nutrients, electrolytes, and water by performing four functions: digestion, absorption, secretion, and motility. After that, I mentioned the different parts of the gastrointestinal tract, said that there is the esophagus, the stomach, small intestines, which includes duodenum, jejunum, and ileum, and then the large intestines, which include cecum, colon, and rectum. After that, I talked about the different layers of the gastrointestinal wall, and I said, in general, the gastrointestinal wall consists of four different layers: mucosa, submucosa, muscularis, and serosa. I mentioned a couple of important points with regards to the submucosa and the serosa. Said that the submucosa is a strong holding layer, so you don't want to injure that or puncture it during intestinal surgery, because that will lead to leakage of intestinal contents, which will basically lead to devastating outcomes such as peritonitis and sometimes death. With regards to the serosa, it's part of the peritoneal lining, and therefore also you have to be very careful, pretty gentle when you handle that layer during surgery. Have to use a lot of fluids to prevent possible adhesion formation. I also mentioned the importance of these layers in doing a myotomy procedure, cutting the muscle layers of the gut, which basically will treat a case that's called esophageal achalasia that I talked about last time. In addition to the layers of the gastrointestinal tract, I also mentioned that there are two ganglionated nerve plexuses that are embedded within the layers of the gastrointestinal tract. These two ganglionated plexuses are the myenteric plexus, located between the inner circular and the outer longitudinal muscle layers, and the submucosal plexus, which is located underneath the submucosa. Both of these nerve plexuses will form what we refer to as the enteric nervous system, or the ENS. I also mentioned that in order for the gastrointestinal tract to perform the four functions: digestion, absorption, secretion, and motility, you have to have a control system that orchestrates these functions together. Again, I gave the example that you don't want to eat a burger and then go to the restroom at the same time. So you have to take your time to digest it, absorb the best of it into your systems, and then get rid of the waste product. And I mentioned that the gut does have such control system to orchestrate these functions that is divided into two parts. This control system is divided into two parts. One that's located within the layers of the gastrointestinal tract, and we call that intrinsic control system, and then one that's located outside the gut or outside the gastrointestinal tract, and we call that the extrinsic control system. Now, each of these systems control the functions of the gut, but also it contains nerves and endocrine secretions. Each of these systems, the extrinsic and the intrinsic, contains or consists of nerves and endocrine secretions. Today, I will talk about the very first part of the intrinsic control system of the gastrointestinal tract, and these are the nerves of the gut that controls the various gut functions. Okay, so the gut function: digestion, absorption, secretion, and motility, are controlled by nerves within the layers of the gastrointestinal tract these nerves called collectively the enteric nervous system. For short, we call them ENS. Now, the enteric nervous system consists of two ganglionated nerve plexuses, a myenteric plexus that's located between or sandwiched between the inner circular muscle layer and the outer longitudinal muscle layer of the gastrointestinal wall. The second plexus is called submucosal plexus. Here in this picture, it's abbreviated as SMP, and it is located underneath the submucosa. So two ganglionated plexuses, myenteric and submucosal, form the enteric nervous system of the gastrointestinal tract. The enteric nervous system is part of the autonomic nervous system, which means that it can function independently, meaning that when you sever or when you cut the nerves that connects the gut to the central nervous system, the brain and the spinal cord, still the gut can function. Why? Because the gut contains its own brain. This brain is called the enteric nervous system. As a matter of fact, some people, some anatomists will refer to this as the second brain, the enteric nervous system, the second brain. Okay, so this is part of the autonomic nervous system. Now, in addition to that, you have to know two main facts about the enteric nervous system, or the ENS. One, the enteric neurons have a pretty unique characteristic. We will talk about that in a second. The enteric neuron contains what we will refer to as veracosities. The second thing about the enteric nervous system is that it consists of hundreds of millions of neurons. Depending on the species, sometimes it's equal to the amount of your neurons in the spinal cord. The number of neurons in the enteric nervous system are equal to the number of neurons in your spinal cord, again depending on the species. So let's attack each of these points. First, the unique characteristic of the enteric neuron, and then the huge number of enteric neurons and how we're going to understand them better. First, let's go to the veracosities. The unique characteristic of the enteric neurons that does not exist in regular neurons. In the upper portion of this picture, you will see a blue neuron, general neuron that has a cell body, an axon, and a presynaptic terminal that secretes the neurotransmitter to an affected organ, like a muscle, like whatever. Now, in the bottom picture, you will see a green neuron. Same structure, cell body, axon, and presynaptic terminal, but also you will see swellings or bulges along the axon. These bulges or these swellings are called veracosities. The importance of these veracosities is that they release neurotransmitters along the length of the enteric neuron. These vercosities are not present in the regular neuron, the blue one in this picture. They only exist in the enteric neurons, which is the green neuron in this picture. And the importance of this structure in the enteric neuron is that neurotransmitters can be released from a very long distance onto a pretty long distance. So along the axon of the enteric neuron, there are bulges that releases the neurotransmitter, and along these axons, you will have muscles that will be affected by these neurotransmitters, which makes sense for the gut because the length of the gut is sometimes 20 and 25 meters, and therefore you need a transmitter that's released a long distance compared to just one spot like the somatic neurons. So this is the importance of the enteric neuronal vercosities, is that they release the enteric neuron along the axon. The whole length of the axon can release a neurotransmitter, which basically affect larger number of smooth muscles in the gut wall. That is a very important characteristic in the enteric neuron. That's why the gut can contract in higher lengths than just a regular muscle. So regular muscle can contract and relax, whereas in the gut, you will see that contraction of the smooth muscles of the gut can take place in a long distance or for a long period of time. And that is due to the vercosities. So this is the first part about the enteric neurons, their unique characteristic in containing what we call vercosities. The other thing I mentioned is the number of enteric neurons, which are hundreds of millions of neurons. And how is that important to us? It's important because we need to understand the function of these neurons. And it is near impossible to understand these functions and what does the enteric neurons do without categorizing these neurons. We have to simplify them so we can understand them. And the way we simplify understanding the enteric neurons is by putting them in categories based on their shape, the shape of cell bodies, based on the electrical activities that they produce, or based on the neurotransmitters that they contain. These three criteria are called morphology, electrophysiology, and chemical coding. Again, morphology is categorizing, putting certain enteric neurons in one category based on the shape of their cell bodies. And we will see examples of that. The second criteria is based on their electrical activity or electrophysiology, the electrophysiological properties of these enteric neurons. Again, some of them are similar, and that's why we put them in one category. The third one is based on the neurotransmitters that they contain in these cell bodies. And based on that, we will put them in one category. And I'll give you example for each of these cases: the morphology, the electrophysiology, and the chemical coding. In 1896 and in 1899, a Russian histologist by the name of Alexander Dogil, shown here in this picture, published three papers identifying shapes of enteric neurons. Cajal named these enteric neurons based on their shape. He called them Cajal type 1, Cajal type 2, Cajal type 3, and so on. And until today, we follow basically his classification. This is a photomicrograph of enteric neurons that are labeled to show the cell bodies and the dendrites. In A, there are large, round, or oval cell bodies with one or maybe two long dendrites. Looks like a tail here in the picture. In B, same, larger cell bodies with one or two long dendrites. In D, the same thing, large, rounded, or oval cell bodies with one or possibly two long dendrites. In C, however, you will see small cell bodies with one, two, possibly three short, very short dendrites. Now, A, B, and D represent Cajal type 2 neurons. C represents Cajal type 1 neurons. So based on this photomicrograph, we will say that we have two types of enteric neurons. Cajal type 1, characterized by small cell bodies and multiple short dendrites, and Cajal type 2 new enteric neurons, which are characterized by a large rounded or oval cell bodies with one or possibly two long dendrites. So these are two major categories, Cajal type 1 and Cajal type 2. There are other classifications, there are other morphologies, but this is the simplest form that I can put these enteric neurons in so we can understand them and follow them better, so we can understand their function. Now, so this is the first criteria based on the morphology of the cell bodies of the enteric neurons, Cajal type 1, Cajal type 2. The second criteria is what? Is electrophysiology, based on the electrical properties of the cell bodies of the enteric neurons. What electrical properties do they show when we record from them? These are two of the recordings obtained from enteric neurons. The bottom one is called an S type. S stands for synaptic, and as shown in the recording, the electrical activity lasts for 10 milliseconds all the way to 0.5 seconds. The second type of enteric neurons is called AH type, and that is after hyperpolarization. and the recording in this type of neuron will last for 20 seconds. So the S type, the electrical activity will last for 10 milliseconds all the way to 0.5 seconds, and the AH neurons will last for 20 seconds. They will last for a pretty long period of time. So two types of enteric neurons, S and AH. It happened that the S type, morphology-wise, is a Dogeal type 1 neuron, whereas the AH neuron is a Dogeal type 2 neuron. So the Dogeal type 1 is an S type, and the Dogeal type 2 is an AH type. Now again, there's multiple differences in these morphologies and this electrophysiological properties, but I am mentioning these for simplification only to understand the physiology of these neurons. But there are so many variations. So we talked about two criteria by which we will understand the enteric neurons better. The first one is based on their morphology. The second one is based on their electrical properties. The third criteria is based on their chemical contents, which means what neurotransmitters do they contain. We have four basic neurotransmitters that we need to remember. The two in yellow on the left-hand side of this picture are vasoactive intestinal peptide, VIP, and NOS, nitric oxide. And both of these are inhibitory motor neurons. So the neurons that secrete VIP and NOS are inhibitory in nature, whereas the green neurons on the right-hand side of this picture are substance P and acetylcholine, abbreviated as ACH. Substance P and acetylcholine are excitatory motor neurons. So again, we have four types of neurotransmitters. This becomes very important when we talk about the contraction and the motility patterns in the gastrointestinal tract. What type of neurotransmitters are being released? We have two inhibitory motor neurons, neurotransmitters, VIP or vasoactive intestinal peptide, and NOS or nitric oxide. And we have two excitatory motor neurons that release substance P and acetylcholine. So four neurotransmitters, VIP and nitric oxide for inhibitory, substance P and acetylcholine for excitatory. So these are the three criteria that will help us understand the enteric neurons better, based on the shape, based on the electrical activity or properties and based on the chemical coding. Now, here I would like to expand a little bit on the chemical coding. We will talk about it more when we talk about the motility of the gut, when we go to special physiology, special gastrointestinal physiology. As I mentioned earlier, I divided the course into two parts: general gastrointestinal physiology and special gastrointestinal physiology. Now we are in the general. We are only doing the basics. The special, we will talk about the specific functions of the gut, the digestion, absorption, secretion, and motility. And in that part, the motility part, I will be talking about the neurotransmitters more and what do they do. But here, just to illustrate the point, what do they do? We have the pancreas and the gallbladder that attached to the upper portion of the duodenum, and they release their secretions through a common duct. In this case, sometimes two little ducts, depending on the species of the animal, but in this case, it's through one common duct. This duct is guarded by a sphincter. It's called the sphincter of Oddi. When this sphincter is relaxed, it's open. That means bile secretions can go into the duodenum, or pancreatic secretions can go into the duodenum. When this sphincter is contracted, on the other hand, there's no secretions coming neither from the gallbladder nor from the pancreas. Now, if we take a sample of this sphincter to see what type of neuronal control does it have, we will see the type of neurons, and this study, by the way, was published by Gary Ma and Kennedy, I think in 1998. Gary and Kennedy are at the University of Vermont. They found that these types of enteric neurons that guard this sphincter are Dogiel type 1, inhibitory motor neuron, and they contain nitric oxide as the neurotransmitter. They call them inhibitory postsynaptic potentials. We'll talk about that later. But now we need to focus on the 3 points that relates to the criteria that we utilized to understand the enteric neurons. Morphology, so these are Dogiel type 1, meaning that they are small and have short dendrites, 1, 2, or 3 short dendrites. These are inhibitory motor neuron, which means that they are S-type. This is the electrophysiological properties of these nerves. And they contain the neurotransmitter nitric oxide, which is basically an inhibitory neurotransmitter. Now, you can understand how the 3 criteria help in identifying what type of enteric neurons we are working with. Meaning that if I take a biopsy and examine it, because my patient is having a problem in digesting fat, for example, that means the sphincter is not being relaxed enough, maybe. This is one of the scenarios, by the way. We'll see that the inhibitory neurons, which are the NOS neurons, are not relaxing. They're not functioning. They're not inhibiting the muscle. And that's how you can have part of the diagnosis. So you'll understand what the problem is based on the shape of these neurons, based on the electrical physiology of these neurons, and based on the content of these neurons. Another example, utilizing the enteric neurons and their classification, is their role in contraction, in the peristaltic movement of the gut. In any part of the gut, you have two segments. Any part of the gut will contain these two segments. One is toward the oral cavity, we call that ORAD, and one that's going toward the anus, and we call that the aborad. So two segments. Remember that we've talked about two muscle layers in the gut, an inner circular, which is the red one in this case, and an outer longitudinal, which is the green one in this case. Now remember, between them there is the myenteric plexus, which is part of the enteric nervous system. In order for you to have the peristaltic movement, at one point you have to have contraction of the circular muscle and relaxation of the longitudinal muscle in order for you to accommodate the coming food into that segment. Let's explain it more. I mentioned a second ago that in any part of the gastrointestinal tract, you have two segments. What are these two segments? One that's called a propulsive segment, a segment that pushes the food in the anal direction, and a receptive segment, a segment that receives that food that was pushed by the propulsive segment. At one point, you have a contraction of that circular muscle where the green line is making this distinction. At that one specific point, you have a contraction of the circular and a relaxation of the longitudinal at the exact same spot. That's for the propulsive segment. Now, when that propulsive segment pushes the food into the anal direction, you will have a relaxation of that circular muscle and the contraction of that longitudinal muscle. That's how the food is being received by the receptive segment. And when this process is being repeated at every single length of the gut, that's what creates what we call the peristaltic movement. At every single spot, we have to understand what neurotransmitters are being utilized. Meaning that if we're talking about contraction, we're talking about substance P and acetylcholine. If we're talking about relaxation, we're talking about BIP and nitric oxide. And based on this, we can determine which neurotransmitter is being utilized at every spot in the gastrointestinal tract. Again, we will come back to this picture when we talk about the motility of the gastrointestinal tract. This is, again, an example to illustrate to you the importance of the criteria that we utilized to understand the enteric nervous system based on their shape, morphology, the shape of the cell bodies, based on their electrical properties or electrophysiology, and also based on the neurotransmitters that they release, either inhibitory or excitatory. If they are inhibitory, then they must be VIP and nitric oxide. If they are excitatory, then they must be substance P and acetylcholine. This happens at every single point along the 23 to 26 meters of the gastrointestinal tract. You have two segments, propulsive and receptive. A propulsive pushes the food to the anal direction into a receptive segment. So the propulsive segment pushes the food to the receptive segment. In order for this to happen, the propulsive segment at that one point I'm talking about, the green line in this picture, there's a contraction of the circular muscle, which means acetylcholine and substance P are in action, and a relaxation in the longitudinal muscle layer, which means VIP and nitric oxide are in action in this propulsive segment. Now, when we go to the receptive segment, we have relaxation of the circular muscle, which means VIP and nitric oxide are in action, whereas you have contraction of the longitudinal muscle, which you have substance P and acetylcholine in action. That's how the peristaltic movement is being generated. Okay, so far I've talked about the three criteria that simplify understanding the enteric neurons because of their high numbers. We have the three criteria: the morphology, electrophysiology, and chemical coding. This basically all about the enteric nervous system. But there's also another part that is equally important to the enteric nervous system in the guts. It's a specialized cell that can act as a pacemaker in the gut, which means that it can generate electrical activity on its own without any stimulation. Just like the SA node and the AB node in the heart, they can generate electrical activity that makes the heart contract. Let's talk about this component that makes the control of the various gut functions take place in addition to the enteric nervous system that we just talked about, the myenteric and the submucosal plexus. This is a picture of a Spanish scientist called Santiago Ramona Cajal. For his discoveries, he shared Nobel Prize with Camillo Golgi in 1906. I think you're familiar with the Golgi apparatus in the cell. Camillo Golgi is the Italian scientist who discovered this Golgi apparatus, the energy generator in the cell. Now, what did Cajal do? Cajal discovered a very important type of cells in the intestinal walls, and he called them the interstitial cells of Cajal, or ICCs for short. Again, these are specialized cells that contains a triangular cell body and 2 or 3 long contractile processes attached, of course, to this cell body. Now, these contractile processes reach all the way to smooth muscle cells in the gut wall and stimulate them to contract. As I mentioned earlier, these ICCs are the pacemakers of the gut. Why they are so special? They are so special because they can generate electrical activity on their own. They don't need any external stimulation to stimulate them to contract. They generate the contractile movement with no external activation. Very important cells, very important cells. And they are distributed all over the gastrointestinal tract. Let's see how they are distributed. This is a photomicrograph of gut wall in mice. This is from a paper of Shane Saunders and his colleagues at the medical school in Nevada. What they did is they did double labeling, immunohistochemistry, double labeling of certain neurotransmitters along with the ICC markers. The ICC marker is a protein that's called c-KIT, shown here in the very first pictures to the left-hand side of this photomicrograph, denoted in C and F. It's the green, like immunochemistry, the KIT, like immunochemistry. This is c-KIT, which is the protein that labels the ICCs, meaning that if you don't have the green, then you don't have the ICCs. You don't have the protein for the ICCs, which means that the cells are not present, which means ICCs are not present. In the middle picture, it's labeling V-acetylcholine transferase, which is a marker for acetylcholine. Again, the excitatory motor neurons. You remember the excitatory neurotransmitters, substance P and acetylcholine, and the inhibitory motor neurons, which are NOS and VIP. Well, here they picked one, the acetylcholine for the excitatory, and the NOS in G for the inhibitory. And they labeled. They did immunohistochemistry for VCHAT and NOS, and you can see them in red. They took the same picture, the same section of the gut, and they put the antibodies for c-KIT on the same section as the VCHAT, and also the c-KIT antibodies on the same tissue, the exact same tissue, with the NOS. And they took pictures with different wavelengths to show different colors, but it's the same exact spot of the same exact tissue. So they did not use different tissues. This is the same exact tissue. All what they did is they changed the filters to see the different colors of the proteins that they stained. So in E and H, you can see that the green, which is ICCs, are basically all over the place in the two pictures, between the red in the upper picture, which is the VCHAT, which means that ICCs are co-localized almost 100% with the acetylcholine neurons. 100% co-localized. They're beside them. They're neighbors. And in H, you will see the same exact double labeling for the c-KIT and NOS. So again, the ICCs are localized exactly beside the inhibitory motor neurons. The two pictures, E and H, will show you that the ICCs are co-localized almost 100% with both the excitatory as well as the inhibitory motor neurons. How do we know that? Because the green, which is the c-KIT label, is basically present in both pictures between all the red lines of the VCHAT and NOS. That means they're co-localized 100%, as can be seen from this picture. So if they are co-localized with these neurons, that means they can work with them 100%. And if the ICCs are capable of generating electrical activity on their own, that means that they can stimulate the enteric neurons, both excitatory and inhibitory, at any time. Remember the contractile processes that projects from the cell bodies of the ICCs in the previous slide. So what's this electrical activity of the ICCs? What do they do? Well, when we record from the ICCs, we will see a number of phases. We have a resting phase, we have a rising phase, we have a plateau phase, and we have a falling phase. So 4 phases: resting, rising, plateau, and falling. Those phases are again the resting, the first phase, or we call the depolarization, rising phase, which is calcium gets inside the cell. The third phase is the plateau phase. And then we have phase 4, or repolarization. falling phase, potassium out. So resting, rising, plateau, and falling. I highlighted the most important phase in this slide, and that is the plateau phase. The plateau phase is extremely important. The plateau phase is extremely important because contraction of any smooth muscle in the gut will not happen unless an action potential is coming from the enteric neuron will meet that plateau phase coming from the ICC. That's when contraction in the gut will take place. Again, you have two requirements for a contraction to take place. You have to have, it's a must, to have an action potential that's coming from the enteric nerve, and you have to have a plateau phase coming from the slow wave, which is basically the whole electrical activity that's recording from the ICC. The four phases in the ICC that we talked about, the resting, rising, plateau, and falling, we call that slow wave, and it's generated by the ICC, interstitial cell of Cajal. In order for contraction to take place, we have to have two important things happening at the exact same moment. One, an action potential coming from the enteric nerve, and then a plateau phase coming from the ICC. This how contraction takes place. If any of these requirements is not present, contraction is not going to happen. And you can see in this picture. In the first picture to the left-hand side, you will see a gut that's having an action potential as shown by the recording in the bottom of the picture on the left-hand side, but there is no contraction taking place. On the right-hand side of the picture, you will see contraction is taking place. Why? Because this action potential met with a plateau phase of the slow wave coming from the ICC. So we mentioned that the control system of the gastrointestinal tract consists of two systems, one that's located inside the gut and one that's located outside. And the one that's located inside, we call it extrinsic, and the one that's located outside is called extrinsic. The one that's located inside contains nerves and endocrine secretions, and the one that's located outside the gut also contains nerves and endocrine secretions. And we started in this lecture talking about only one part of this intrinsic control system, and that is the nerves. We talked about the enteric nervous system as well as the interstitial cells of Cajal. And we said that these are very important cells because without them there will be no contraction in the guts because they generate slow waves. Okay, so what happens if we lose these ICCs? What happens if there are loss of ICCs? Meaning that you have a patient with a number of abdominal problems like a patient that's not able to go to the bathroom, having colic all the time, um, having difficulty digesting their food and things like that, whether it's a human or an animal. We need to take a biopsy of a certain part of the guts, either laparoscopically or regular surgery, and we will see that when we stain these cells, when we stain these sections with ice with C-kit, the one that I showed you in the other slide with the green lights or the green color, you will see that this green line or the green color is not present, which means that C-kit is not being labeled, which means that ICCs are not there. And that is why you have some motility problems like chronic intestinal pseudo-obstructions and smooth muscle tumors. That is why you have loss of ICCs. And the ICCs basically change with the age. They are reduced with age. So the older the animal or the human, the less ICCs you will see. And that's why that may explain, if you will, the difficulty of some older people in going to the bathroom compared to younger uh, uh, individuals or animals, either. So we've talked about all of the components of the intrinsic control system of the gut, the nervous part only. The nervous part. This is the roadmap that we're following in our discussion here. And so far, uh, I've only talked about the nervous part of the intrinsic control system of the gut, which contains the enteric nervous system and the interstitial cells of Cajal. Next time, I will talk about the nerves of the extrinsic nervous system that controls the gut functions, the vagus and the splanchnic nerves.